Okay, so next let's think about the T cell response to cancer and how this goes wrong. So we know from just decades of work that cancer really is evading T cell killing. And how this happens is it's driving the T cells to a state called exhaustion. So normally T cells are circulating in our blood, and we're going to call these naive T cells. And when there's a tumor, the T cells will migrate to the site of the tumor. So for instance, in melanoma, the T cells will migrate to the skin. And these T cells will encounter the cancer cell, which is displaying on its surface this non-self antigen, and they will enter what's called the progenitor state as they become activated. Now, these T cell states then start to transition. So the progenitor state goes to a state that's called the effector state, which is good at killing tumor cells. And then this state, unfortunately, transitions over to a state called the terminal exhausted state, which is not so good at killing tumor cells. So these T cell states, they're all considered to be exhausted T cells, but they are exhausted to different degrees. And the T cell states themselves are encoded by gene expression programs. So remember, these are groups of genes that are turned on together in sort of a shared biological process. And these gene expression programs are really analogous to the gene expression programs we'd introduced for cancer when talking about melanogenesis or this epithelial to mesenchymal transition. So similarly, each of these T cell states, the progenitor state, the effector state, and the terminal exhausted state, are encoded by gene expression programs. And measuring these programs will tell us what the T cells are doing. So now let's look at this from a more visual setting. So here we have this diagram where we're going to look at the T cell response to cancer. And on the left y-axis, we have the cancer burden, so how much cancer there is, and that's shown in this brown line. And then on the right y-axis, we have the T cell response in purple. And so again, what happens is a naive T cell will migrate to the site of the tumor as the cancer starts to grow, and it will transition to a progenitor state, and then it will further transition to an effector state. And as it transitions to the effector state, the T cell starts to actually kill the cancer cells. So this is where you get this peak in the cancer burden, and it actually starts to go down. Now, unfortunately, the effector T cell state does not last. It actually starts to transition to this terminal exhausted T cell state, which is weaker at tumor killing. So the T cell response starts to go down. The cancer burden just starts to stabilize. And so you reach this stalemate where the cancer is still there and the T cells aren't quite able to clear out the cancer. So there's a stalemate or détente between the cancer cells and the T cells. So scientists have been studying terminal exhausted T cells for decades, and here are some consensus features about those T cells. So the first is that they have this inability to control the cancer growth. So you don't go all the way to having no cancer, it just sticks around. There's also this loss of the T cell effector killing function. So the T cells are blunted and they're no longer really actively killing the cancer cells in a manner that controls the cancer. There's also these expression of these inhibitory receptor genes, and this is going to be talked about in the cancer immunotherapy section. And then finally, the T cells in the terminal exhausted state have less ability to undergo self-renewal and make more of themselves. Okay, so let's recap what we just learned in the past two slides about T cell exhaustion. So naive T cells are starting in the blood, and then they migrate into the tumor, and they enter into this sort of exhausted set of programs. And the T cells, when they encounter the cancer cell, enter the progenitor state, and then they differentiate or transition into an effector state. And finally, they enter this bad terminal exhausted state. Now, the terminal exhausted state, as we said before, is weak at killing tumor cells, whereas the progenitor and effector states are very strong at killing tumor cells. And finally, there's a last state that the T cells can enter, which is the cycling state. And the cycling state is just the T cells entering the cell cycle and making more copies of themselves as they undergo cell division. So cycling is particularly good, especially for the effector T cells, because these are the ones that are going to go in and kill the cancer. And if you're making more of those effector T cells, it's like having a bigger weapon against the cancer. So what's happening in the cancer environment, again, is that there's something about the cancer and the surrounding tissue environment that's pushing cells away from the progenitor states and these effector states and forcing them into this terminal exhausted T cell state. And so you can think about this as a distribution, right? The T cells can be in any of these states, but in the cancer environment, there's a strong push to be in the terminal exhausted state.